All right, welcome to our Introduction to Cells lab. Here are materials for this week: newspaper, scissors, compound light microscope, toothpick, methyl or methylene blue, glass slide and cover slip, those should be blank, and an onion. Okay, so starting with part one, we're going to take a look at the letter E from a newspaper under the microscope. So cut out the smallest letter E that you can possibly find on your newspaper. And a magazine will not work because the paper is too thick and the light from the microscope won't pass through it. So cut out your E. It should be super tiny. And you're just going to want to put it on your slide. Just like that. And then we're going to put it under the microscope. And you're going to use low power, which on my microscope is 4 times magnification, that lens. And then again, you're going to magnify it by the low power times the eyepiece, which is an additional 10 times. That would be 40 times on low power. Okay, so this is what it looks like under my microscope. I'm starting with the stage all the way at the bottom and using the large knob, the coarse adjustment, to bring the stage up slowly until the newspaper comes into focus. And then I'll use the smaller knob, the fine focus, to make it nice and crisp once it's as close as we can get it on the coarse adjustment. And you'll notice that you can see the newsprint E, and it is upside down. And that's because the lenses on the microscope flip objects over. Okay, on to part 2. We're going to look at human cheek cells. So you're going to take a toothpick and rub it on the inside of your cheek until it's wet and covered with some of those dead skin cells on the inside of your cheek. And you're going to rub it on the slide. Should be a little bit of a film on it. And then you're going to put your cover slip on, and we will create a wet mount, a stained wet mount of these cheek cells, because they are clear otherwise and pretty hard to see. You can certainly see them if you don't have stain, but it's going to be a little bit harder. So we use the methyl blue or methylene blue, and you're just going to put 1 drop on one side of the cover slip, and you're going to tilt your slide, and with a paper towel you are going to draw that stain through the specimen, and then the paper towel will absorb the extra stain. If you just plop a drop of stain on top of your cheek cell specimen, it will flood the slide and you really won't be able to see anything very well. Things will be moving around because it's just too sloshy and wet. So get as much of that stain on the paper towel as you can, and then you're going to put it under the microscope. And we always start on low power, but this time we'll bring it all the way up to high power. Okay, so we are starting on low power, and again starting with the stage all the way down. And then as you're looking through the eyepiece, you're focusing and bringing that stage up until something comes into focus. And here you'll see our cheek cells, which are stained slightly blue. Yours might be a little bit darker depending on which kind of stain you used and what the concentration is. But so we're going to focus even higher on some of those cheek cells so that we can see them better. So we're switching without adjusting the focus again. We are switching to the medium power. And wait till my camera adjusts here. Okay, and you can use the coarse focus knob if you need to. You shouldn't need to move it very much. And fine focus, the smaller knob, to bring that in nice and crisp and clear. And then we're going to move to high power. And on high power, you should only be focusing with the fine focus knob, or you could smash your slide into your high power knob or high power lens and scratch the lens. So here we're looking at a smaller field of view, and you can see that I really can't see any of my cheek cells anymore. So I'm going to go back to medium power and make sure that I have my cheek cell right in the middle of my field of view, so that way when we switch to high power again, you should be able to see it. So we're going to move that right back to the middle. Hard to move things gently when you're on such a high magnification. Okay, now we got that in the middle, and I'm going to switch back to high power. And now we can see our cells a little bit better. I'm going to move it around again, see if we can get to see them a little bit more clearly. And I'm just moving the slide. This microscope I'm using does not have a stage or slide adjustment knob, so I have to use my fingers and shift things back and forth. But now you can see that cheek cell, and I'm going to use the fine focus to make it nice and crisp and clear. And then we'll take a look at some of the structures that we can see. Okay, so you can see the cell membrane, which is the outer part of the cell, and then the nucleus, which is that circle structure in the middle, and then some tiny little dots of bacteria, which we can't see closer than this with this microscope. Okay, part 3, we're going to look at some onion cells. So your lab says to use a red onion. You can use a white onion too. Either one of those would work. Red onion will give you a little bit more color. And you're going to want to peel off the thinnest possible piece. You can see how thin that piece is that I'm using. And you're just going to lay it right on your clean slide. Make sure you've rinsed your slide off from all that stain. And you're going to flatten it out just so that you can see only one layer of onion there thick. We don't want it to be rolled up. And we're going to look at that under the microscope, starting again on low power. And this time you're going to use low power and medium power. Okay, again, this is what I'm looking at under the microscope with the stage all the way down on low power, bringing it up slowly until the specimen comes into focus. And there it is. Those are our onion cells. And those little circles are air bubbles. And then there's some other miscellaneous debris on there, but we want to look at those boxes. And there's the air bubbles again. Okay, so you can see those boxes there are the cell walls of the onion, and the cytoplasm then is on the inside of that. And I'm actually going to adjust the diaphragm of my microscope here to make this a little bit darker because I was getting too much light through the microscope. And the diaphragm is under the stage. You should have a little dial, and that lets less light through. So we're going to refocus here. Sometimes you can see the nuclei in the onion cells. This time it doesn't seem to be showing up very clearly, but you should be able to label what you see. So here's a labeled picture of onion cells. This is one I took a little bit earlier, so you can actually see the nuclei a little better on this one. But you do need to label the cytoplasm and the cell walls.